Oh. Click on children. Okay. I'm just lost. Yep, good. Okay. But then it comes back. No, that's how it was before, but then it came back for some reason.
Good morning, everybody. I'd like to welcome everybody to the IAE seminar for this week. I'd like to acknowledge the Ngunnawal people as the traditional owners of the country on which we work, where the seminar is presented, their continuing connection to country, especially water, and I acknowledge leaders past, present and emerging. So today, it gives me great pleasure to introduce Alicia Holland. Alicia's from La Trobe University. She works in a whole range of areas. It's amazing, actually, when she describes what she works on. But today, I think she's going to focus her talk on dissolved organic matter and its interaction with chemicals in aquatic environments. Yeah, without further ado, I'll hand you over to Alicia. Thank you.
Thanks, Rod. I'm not going to include any chemicals today. I'm going to talk about microbial communities and productivity and the role of dissolved organic matter. Firstly, I'd like to acknowledge the Ngunnawal people on the traditional owners of the land that I'm giving this seminar today, and I acknowledge the traditional owners past, present and emerging. Yeah, so as I said, I'm going to talk about dissolved organic matter, and basically I'm going to focus on how it can enhance productivity within freshwater ecosystems. But firstly, before I even start speaking, I'd like to acknowledge the people that actually do the work that I'm going to present today, especially in regards to the case study. So I have two of my PhD students here. I have Saman Akia, who did most of the work for case study 1, and Francois Etienne Sylvain, who did most of the work for the case study 3 that I'll be presenting today. That thing still, sorry, up there. It's going to disappear. That one? Sorry about that. I'll have to keep doing that. And I hope everyone can hear me online as well. So firstly, I thought I'll give you a brief background of what actually is dissolved organic matter and what I'm talking about today and what I mean when I mention dissolved organic matter. And I might refer to it as DOM as I talk. So that's what I'll be talking about. So basically, it's formed by the decomposition of animal and plant matter. And you might have walked down the street and noticed these stains on the various footpaths of leaves. This is basically the dissolved organic matter leaching out of the leaf tissue as it decomposes. You might also go for a walk around Lake Burley Griffin and notice the algal blooms, and then you can see that it might have this sludgy looking stuff on the outside, and this is the algae material being broken down, decomposing. You might also notice that when leaves or sticks are in a puddle, the water might end up with a quite tinge to the water, and this is the dissolved organic matter that's leaching out of that material as well. But I clicked on it, it's not going to skip. But it's formed by the decomposition of animal and plant material. Microbes can also release dissolved organic matter into their fresh waters. And here I have an example of Alexandrium, which is a toxic algae or red tide algae that you might know it by. And what we looked at is the amount of dissolved organic matter or dissolved organic carbon, in this case, released by this algae. And what we figured out is that basically it releases about 60 nanograms of dissolved organic carbon per cell into the water under various conditions. And algae, different material will leach different amounts of dissolved organic matter as they decompose, and different algae will release different amounts as exudates, and that is also. And here I have an example of different leaching experiments that we did on. So we went out and collected different matter from floodplains and looked at how much dissolved organic matter is leached in the form of dissolved organic carbon from the various fractions. And what you can see here is that leaves tend to leach a lot more dissolved organic matter than, say, the other fractions such as plants or sticks, for example. And dissolved organic matter is ubiquitous everywhere, especially in fresh waters, and it tends to consist of dissolved organic carbon, nitrogen, phosphorus, and also some sulfur components. But the organic nitrogen and the organic phosphorus tend to make up a smaller proportion of the organic matter compared to the organic carbon. And I noticed I left out my x-axis, but basically these are different sites. And these are, here you can see we've got organic carbon in milligrams per litre. We've got organic nitrogen, which is about a factor of 10 less than the carbon component. So we're looking at micrograms per litre here, and it's the same with dissolved organic phosphorus. Waterways will contain the same dissolved organic matter. So it's made up of all these different molecules. So even within the same system, the dissolved organic matter will change depending on its source, its composition, which depends on where the dissolved organic matter originally came from. So that consists of like its key components, also differ in the amount of carbon to nitrogen, and also in its characteristics. So how aromatic is the organic matter, what is its molecular weight as well. And this will vary temporally and spatially, even within the same site, across systems and that as well. And I'll give some examples today as I talk. As I mentioned, it's a wide variety of different molecules, so it's very heterogeneous in nature. And as you can see, that it's a very complex mixture of macromolecules. So most of dissolved organic matter in freshwaters tends to be in the form of humic substances, and these can make up 50 to 90% of the organic matter pool. But then we also have carbohydrates, proteins, organic acids, amino acids, ketones, and aldehydes that can also contribute to dissolved organic matter within fresh waters. Method in characterizing dissolved organic matter in fresh water. So different research groups use different methods. There's a wide variety of different techniques in the literature that have been used to characterize dissolved organic matter, but there's no standardized approach. And today I'm just going to focus on two different methods. So basically, dissolved organic matter can be characterized either using optical methods such as absorbance or fluorescent spectroscopy, or it can be characterized using analytical methods such as liquid chromatography or NMR, or other various different, such as FTIR as well. But today I'm going to focus on two main methods when I talk about the organic matter characterization, especially in regards to the carbon. And I'll talk about a third briefly when I talk about nitrogen as well. But the two methods that I'm going to focus on today is the use of fluorescence and absorbance indices, such as fluorescence excitation emission scans. And basically, we take a water sample and we scan it at various excitation emission wavelengths. And basically, depending on the size of the molecule, will depend on where it fluoresces in this. And you end up with this heat map here. So the larger molecules that consist of more humic materials, such as humic acids or fulvic acids, tend to fluoresce at higher emission wavelengths compared to the smaller molecules that might represent more proteinaceous material. So I'm going to talk about that method. I'm also going to talk about liquid chromatography with built-in organic carbon and nitrogen detectors. And basically, this works similar to all liquid chromatography. It's based on mass. It breaks it up into they elute at different times depending on their molecular weight. And basically, it breaks it up into 6 main fractions. So biopolymers, humics, which consists of your humic and fulvic acids, your building blocks, which tend to be your breakdown products of your humic substances, your low molecular weight acids, and your low molecular weight neutrals. And you'll see results from both these two methods. When I bring up the organic nitrogen results, I use liquid chromatography with a mass spectrometry. and I'm characterizing amino acids. And I'll talk about that later. As I mentioned, we can use absorbance and fluorescence indices to indicate different characteristics of dissolved organic matter. And here I've just put a couple of key indices that might be on my graph, just so you don't have seen this briefly before I talk about it. So we have specific UV absorbance indices that can give us an indication of how aromatic the dissolved organic matter is. There's other ones such as the fluorescence index that can give us an indication of source, so whether it's more terrestrially derived or more microbially derived. There's also some that can give us an indication of molecular weight. So given that these won't give us an actual value for aromaticity or an actual value for molecular weight, what they're useful for is to basically, if it's higher or lower, to compare across sites. So if one's higher, it tends to be either more aromatic or higher molecular weight compared to another site, but it won't give you an exact value. Okay, and here comes that thing, hiding my thing again. Okay, so why do I care about dissolved organic matter in fresh waters? Well, basically, it forms the basis of the microbial loop within aquatic ecosystems, and this can then lead up to the higher order consumers. So here's just a model that you can see here. We've got dissolved organic matter coming into the freshwater system. These can either be produced within the system by decomposition of material within the water body, or it can come from outside, so plant leachate, leaves falling in, leaching dissolved organic matter. And this will form the basis of this microbial loop, feeding the bacteria and these mixotrophic algae, and then go on to feed the other organisms. Now I got to click here again. I'm sorry about this. Makes me look unprofessional. Okay. So to the second part of my talk. So I've briefly touched on what is dissolved organic matter. And now I'll focus on microbial communities and why we're interested in it. And I'll briefly provide an example of what we call a priming effect as well, because I'll talk about that as I go along in my case studies. So basically, a priming effect is where the addition of one form of dissolved organic matter to a pool of another. So say we had a lake or a river that had mostly terrestrially derived dissolved organic matter, heaps of leaf litter, most of it's really humic, high molecular weight dissolved organic matter, and will have a certain microbial community. Then we have a river that's flowing into this lake that has, for example, more autotrophic production in there, has a higher microbial dissolved organic matter. And then the mixing of these two pools together can sometimes lead to enhanced productivity outcomes. So when I talk about productivity outcomes, today I'm going to talk about the amount of carbon either produced in the form of CO2 or consumed, so CO2 being taken up and algae releasing oxygen into the atmosphere as well. So just so you're aware. And here I've got a conceptual model that I've developed. So here we've got one system where our algae is dominated, we get high autotrophic productivity, heterotrophic pathways not as great. So allophonous DOC, for people not aware of those terms, allophonous means something coming from outside of the system. So in the case of freshwaters, I tend to use this term as terrestrially derived carbon coming into the system. Autochthonous tends to mean produced within. So produced within the freshwater, so microbially derived on, yeah. Another one where we get higher allophonous, so higher terrestrial input into our freshwater, you get higher bacteria, higher heterotrophic productivity or respiration, and the algae signature is not as strong. But when we get the mixture of two, we might get equal or similar because we have the mixture of the two, and then we might get enhanced productivity outcomes for both the autotrophic and the heterotrophic pathways. And I'll give you some examples of that as I talk. Okay, so today I'm going to use 3 case studies, and these are from different systems. And there's different backgrounds to why I actually started investigating this priming effect. And the first one will be in regulated and unregulated system, and we'll talk about dissolved organic matter, also the microbial communities and productivity. And this will be focused in the Tumut-Gubagandra system. The next one is in the Warrego and Darling systems, and then we'll go fly overseas and we'll have a look at what's happening in the Amazon basin as well. So case study 1. So this study came about more from the point of view whether tributary inflows could enhance productivity outcomes in regulated systems. So basically we have the Tumut River here, and if I can show you with my pointer, you have the Tumut River here flowing down, and then you have the different reservoirs here. So Blowering Reservoir is the one above our sites, and then we had sites upstream as well. But then we have this unregulated tributary nearby called the Gubagandra that joins the Tumut River downstream from these reservoirs. So we wanted to know whether this tributary, the Gubagandra, could enhance productivity outcomes downstream of where it joined. I'll focus on just a small proportion of the results. I'll focus on the dissolved organic matter in the microbial communities as well as the productivity results today. But basically what we did is measured concentration and the composition of dissolved organic matter within these systems. And what we found is that the tributary site had high concentrations of DOC. So tributary is TD. I don't know, my students should have chose better acronyms, but that's okay. Contained higher amounts of organic carbon, organic phosphorus, and organic nitrogen than the regulated, which is this MD, and the confluent site. When we looked at the composition of the DOM, we saw that the tributary contained a composition of dissolved organic matter that's really similar to the upstream site within the Tumut, so above those reservoirs. So before the Tumut River flowed into Blowering Dam, the organic matter is similar to the tributary that didn't have a reservoir. But the processes that occur within the reservoir changes the types of organic matter, and you get a different types of organic matter. Basically, you see smaller molecules coming out that are produced within that reservoir. So more microbially derived DOM compared to the more terrestrial, larger molecules, such as the humic-like components that you find in the upstream and the tributary. We also did organic nitrogen and looked at the amino acid composition of the organic nitrogen, but that signature wasn't that strong in this system. There's slight differences, but really they didn't fall apart. But what we noticed is that even though the confluence was receiving, if I jump back here, was receiving organic matter from the reservoir also and the tributary, the reservoir still had the strongest signature towards that confluence. So if you have a look here, the confluence is the purple, and yes, it's sitting between the tributary and the regulated system, but it's more closely related to the regulated system. And that's because under base flow conditions, the regulated system forms the majority of the flow within that waterway. And we saw that here as well. So if you look here, the confluence is sitting somewhere in between the regulated and the tributary. Biofilm communities, and what we saw is that, yes, they contain different biofilm communities. Basically, tributaries blue, regulates green, confluence is red. Basically, the tributary contained a unique fungi, algae, and bacterial community, whereas the confluence and regulated contain more similar. This PE is, so this is a bit intuitive. D4 is day 4, P is pre-event, so pre a storm event. D4 is 4 days after the event. D18 is 18 days after the event. So we thought that during a storm event, you're going to get higher flows of this tributary influence in microbial communities. And what you see, you do see some transition, but during day 4, for some reason, the confluence was actually similar to the regulated. And when we link that to DOM composition, it suggests that the different types of organic matter may play a part in the different microbial communities. But what does this mean for productivity? And to assess productivity, we use chamber experiments because in these systems, they're highly aerated, like dissolved oxygen's basically 100% most of the time. They're really riffled systems. It's hard to log metabolism over a long period of time. So we decided to do pelagic and benthic chambers, and there's our chambers up there. And basically we seeded tiles out in the rivers for 4 weeks before this experiment. We had light and dark, with and without tiles to mimic the benthic and pelagic. And what we saw here is that in regards to, so we also have autotrophic and heterotrophic. So black's heterotrophic in the dark, red's indicative of autotrophic production in the light. And this is oxygen produced or consumed on the Y. And what we saw is that the confluence had higher autotrophic as well as heterotrophic production, even though it was more similar to the regulated site. And this was a factor greater than what you would expect by the mixing of the two organic matter pools. So this suggests a priming effect is happening under this tributary. For other parameters, so we measured biomass, chlorophyll A, total biofilm, and the confluence always had higher microbial biomass. So that was the first study that we investigated this in. And recently I had a chat with Sarah and Micah, and we were talking about food webs and metabolism, and that I'm like, oh, it'd be cool if we could look at this within a different system. And as part of the MERB project that some of you work on, Sarah and I decided we'll look at it in the Warrego and the Darling rivers. So I'll talk about just organic matter, concentration and composition again, as well as microbial communities, but this time I'll be talking about the ones floating around the pelagic microbes rather than the biofilms. And basically we went out and sampled all these sites, a total of 8 sites within the Darling and the Warrego, and you see differences in DOC concentrations between different sites and differences between months sometimes. And mostly they were quite similar, but there is differences. But the Western floodplain always had less DOC as well. We also did the characterization of the organic matter. And what we saw is basically the Darling had more terrestrially derived organic matter, so more humic-like materials, so larger molecular weight, more aromatic, coming from that terrestrial environment compared to the Warrego. And the Warrego and the Western floodplains had more microbially derived DOM, so this higher protein-like signature. And when we put it together with the LCOCD results and those absorbance indices and put it all together as this mess of a PCA, you can tell that they pull apart. Western floodplains blue, Warrego's green, Darling here. And basically this says that the Darling contains more, like I said, terrestrially derived DOM, high molecular weight, more aromatic compared to Western floodplains. It's more microbially derived, and Warrego is somewhere in between. And here we did it by site as well and get site differences. And what I want you to note, and I'll come back to it as I talk, we did chamber experiments at 3 sites. At Ross Billabong sits here, Bawera sits here, and Dick's Dam sits there. So Ross sits more closely aligned with the Darling than the Warrego, than the other 2 Warrego sites. And I'll touch on that as we go further along. But what did we find out about the microbial communities? And I've grouped these based on phylum or like a high level grouping. You could do species. We had that data, so we used a metabarcoding approach where we filtered the water and extracted the DNA. We used 3 primers to target both the fungal, the algal, and the bacterial communities, and I've broken those apart here. What you see is you get differences in the microbial communities between the different waterways as well. So here, for example, for algae, you can see diatoms dominate the Darling compared to the Warrego, whereas the Warrego, you have this Orchophyta, which tends to be a golden brown algae, more abundant within that system. And when I say abundant, these are reads, so it's not true abundance. It's just indicative of what might be happening. In fungi, you get similar differences. So chitta fungi is higher in the Darling compared to the other site. And then when we look at the bacteria, you get high amounts of this proteobacteria within the Darling compared to the Western floodplains and the Warrego, which tend to have more of this, especially in the Western floodplains, this bacteriodex bacteria. And I'll talk more about it probably in the Amazon, but basically proteobacteria has the ability to break down humified material, whereas Bacteroidetes does not. And these need more labile dissolved organic matter to actually break down and utilize as an energy source. We did an RDA to link it with the communities with the composition and basic water quality as well. So all water quality, nutrients, phys chem, was placed in this RDA to determine which ones might be driving it. And basically majority were organic matter characteristics that pulled apart the communities. And basically it was the more humified DOM in the Darling led to differences compared to the more proteination microbial derived DOM within the Warrego and the Western floodplains. Similar to what we did in the Tumut Bagandra, we did our chamber experiment. So here's our racks that we had out. We put them in the different containers here, measured oxygen before and after. We had pelagic and benthic, so we had containers with and without the tiles. And we also did light and dark as well. And basically what we saw, so remember I mentioned the 3 sites, Bawera, Dicksdam and Ross. Bawera and Dicksdam were true Warrego sites that sat over the far side of the PCA. Ross sat more similar to the Darling because it receives water from both the Warrego and the Darling systems. And what we saw is that you get higher productivity within the Ross Billabong here. And the colours represent benthic and pelagic. And we get a stronger benthic production signature within this system as well. And these 2 sites, so we plan to directly compare the Darling with the Warrego with productivity. However, when we went out to sample, the Darling was in flood and we couldn't access our racks. They were out way. We would have had to swim for ages to get our racks. So that's why we ended up with the 2 Warrego sites and a mixture of Ross Billabong, which has the 2 waters. But this also suggests that that may be a priming effect of the mixture of the 2 organic matter sources was happening here. And here's our PCA again. So you can see, like I said, it was more closely related with the Darling than to the Warrego site. And when we looked at the microbial community, so Ross Billabong's on the end, what we saw is that it contained a more diverse microbial community compared to the other 2 sites as well. And this may suggest why we're getting an enhanced productivity within this system. Okay, so lastly, I'm going to fly you across the other side of the world. We're going to go to the Amazon and we're going to look at the different water types that we have within the Amazon basin. We have, basically black water rivers. So all of you, they look like Coca-Cola, basically like that. They're highly tannin stained, high in organic matter. We have our white water turbid rivers, such as the Amazon River or the Rio Solomois, which look more like this. And then we have our clear water rivers or the Rio, such as the Rio Tapajós. And what we went out is we went out and we sampled 15 different sites for organic matter composition. We did bacteria plankton as well. And this is only part of a larger project. We also did fish microbiomes as on top of this, but I'll just talk about the free living ones in the water and the water chemistry today. But what we saw is, like the other sites, you get clear separation of organic matter based on characteristics, based on the water type as well. So you have black water, I think is the red, more humified organic matter, which makes sense because it's that dark color, compared to your clear water, which is more proteination, microbially derived organic matter. So when we collected the water samples, we used a metabarcoding approach, but we also did envirotranscriptomics as well. So we also immediately preserved the samples in an RNA buffer and then also looked at what genes were being expressed within the bacterial plankton community at that time as well. And what we saw is you get differences in the structure of the microbial community. So we're just talking about bacteria here between the different river site types. You also get differences in the function of the different waterways. And you also get a difference in what's being transcribed or upregulated in that community during that time. And basically, when we did all different types of stats, or when I say we, I mean my PhD student, Francois, did all this work. I'm just taking credit for his great job. When we did the stats, we related it to the amount of humic, dissolved organic matter. We could see clear relationships here with the networks here. And like I mentioned before, the proteobacteria contain the genes that can break down the humified material. He's gone to a higher level than I identified previously. So there's polynuclear bacteria, the proteobacteria, are all within that group that have the ability to break down this humified DOM. So we took this huge amount of data from the metabarcoding. We also took the transcriptome, so what genes we picked up that were upregulated. And then we sort of scanned the literature for the genomes of each bacteria that we could possibly find, created a database, and then did correlations between the genes we actually measured in the water, the taxa we found, and whether they had that ability or not. And what we found, and we correlated this to humic DOM, is that the bacteria, these three mainly, mostly polynuclear bacter, were highly correlated with these genes that are involved in the decomposition of terrestrially derived humic material. And this is likely why they're more abundant in the black water environments compared to the white water and the clear water environments. So when we look at those three case studies together, what we can see is dissolved organic matter differs spatially and temporally among sites. So we can't just go take a water sample and say, okay, our carbon concentration is this, and expect to get the same outcome as another site that might have the same carbon concentration, because every site slightly differs. And basically the composition of organic matter can range from terrestrially derived high molecular weight humic DOM to really microbially derived, really labile, really bioavailable smaller molecules of DOM. And it could still be the same concentration of carbon in the water. What else? Microbial communities differ both temporally and spatially, and that's suggested to be associated with the changes of organic matter composition. And basically, we have a little bit of evidence, so we still need to collect more from different systems around Australia, that the addition of two different pools of DOMs and the mixture of that DOM may lead to more enhanced productivity outcomes in freshwaters. And I'd like to thank my team at CSIRO and La Trobe University, as well as all my students over the years and my funders, and I will take any questions. Thank you.
Thank you, Alicia. Does anybody have any questions?
Thanks, Lisa. It's really cool stuff. And just looking back to some of the work you were doing with the chambers in the field, do you ever get the situation where it's basically they're nutrient limited rather than light or carbon limited? So you sort of end up with a productivity response in the first hour and then it drops away. Is there any incidence of nutrient limitation in these systems?
Good question. So we leave them out. So in the Warrego and Darling, because of the travel time between sites, we could only leave them out for 4 hours. In the Tumut Gubagandra system, we left them out for 6 hours. We measured the nutrients at the start and at the end, and we didn't see any evidence of nutrient limitation. And even the carbon uptake was only minimal, like could only just you could see that it was lower, but it wasn't statistically different sort of thing during that time period. Did that answer your question?
Alicia, that was really great. I was wondering about the role of fungi and how that fits into stuff. It's not something I've really ever thought too much about, rather than just my bacteria and algae and stuff. And yeah, how that fits into DOM and its place in the food web.
Good question, James. So if I'm correct, you're asking what the role of fungi in decomposition of organic matter or microbial production. So we've only done. So we did fungi in the biofilm, and we did it for the first time in water as well in this study. And we did see the different communities, and basically similar to bacteria, different types of fungi have different genes sort of thing. So some species have the ability to break down humified organic matter, and it's suggested that they actually play a major role in this cycling of that terrestrial organic matter when it enters fresh waters. So along with the proteobacteria, there's also the fungi that let me see here, the ascomycota is especially said to have those enzymes that help break down those large lignin-derived like humic molecules as well. How that actually does it. So when I say dissolved organic matter, it's like basically defined as whatever goes through a 0.45 micron filter. So we do have colloids floating around in the water that the fungi could be living in and feeding off, I guess, as well. So yeah, and the association between the bacteria, fungi and algae is interesting as well, because similar to like us, we have bacteria living on us. Algae have bacteria that feed on them, on the exudates, that have like a symbiotic relationship. Whether there's symbiotic relationships with fungi as well, I'm not 100% sure. And because they're not autotrophic, they probably lead into the higher respiration as well that we see wherever that is. I answered that question. Good. Darren?
Thanks very much, Alicia. Yeah, really interesting. Has there been any like laboratory tests of this priming effect using different DOM sources in Australia?
Yeah, so how the priming effect, it originates from the soil literature. So where different pools of organic matter in the soil has been suggested to increase to enhance productivity. There's been batch experiments done as well, where they spike different organic matter, and the results, some are consistent with a priming effect, some aren't. Same as when there's been reviews done on priming effect in freshwater. Sometimes it's shown, sometimes it's not, or it's not really strongly indicative of a priming effect. So I guess the literature isn't inclusive of whether it's true or not, but I think it depends on the type of the organic matter and the ratios and the system as well, whether you're going to see that priming effect.
Yeah, okay. Like some of the example comes from this flooding period. Could it be?
Which one?
Was this in the case study too? You saw some of that evidence, and I was just
in the
curious.
here.
Yeah, if that's potentially related to this, that it's in flood and you have a lot of different types of.
Yeah,
so that could increase the effect.
The Warrego was in flood as well. So during that period, what happened? So we sampled last year. So after that big flood, we sampled in March and April. And at the time, they were doing work on the dam in the Warrego, so Bewara Dam. So they'll send in the water through the western floodplains into the Warrego as well. So all that was flooded, and the Darling was flooded. So both sites had similar flooding, I guess, inundation, except that where we put, so you can see some of the, even here, we managed to get to it, but in the Darling, it was just too dangerous to send the team out to get the chambers. It's a complete difference.
Cool, thanks.
Me again.
Yeah.
I guess I probably should know this, but I spend more time filling in risk assessments than I do reading papers.
Mm-hmm.
So has there been work done on sort of the effects of some of the stresses into these systems? I think of something like Lake Burley-Griffin, where you've got a big pile of caffeine and antibiotics coming into it, or the urban systems like Rod works on, where you've got lots of zinc and copper. Do you, presumably you would see changes in the microbial communities. Has that been well described?
Not well described. So in a natural setting, not much has been done. Like we get an understanding of single taxa from experiments that we do. And a lot of my work, and that's probably why Rod got confused and said I was going to talk about chemicals, focuses on metals and like algae tolerance and that kind of thing. But from a whole community response, there's not much known, but definitely it will cause changes in the community structure. And I did measure metals in the Warrego and Darling, and the results probably aren't that good, but I want to repeat it just to make sure that it wasn't like a laboratory, like some mistake by the labs, some contamination. So
yeah, we had a PhD student who finished last year who was looking at water going to Lake Burley-Griffin and looking at decomposition rates. And he was finding lots of caffeine, lots of amoxicillin, all of these things coming out of bottom sewage treatment plants, which were halving or
Yeah, so-
even more reducing the decomposition rates.
Yeah, so there's stuff done on like, like you said, decomposition rates, but not really shifts in what's driving that. So they'll go out and measure, like they'll put leaf packs out and look at how quickly they were sort of break down in contaminated environments compared to others and that kind of thing. So
Sounds like my people should talk to your people.
should. Anybody else?
Any more
And
questions?
sorry if I butchered the microbe. It's not really my background. So
Any more questions? Do you want to check the
Online.
chat?
Yeah,
is there any questions online? You can either turn your mic on or type a question.
Last
I
ones
think
in
they're
the
sick of
audience?
listening to me. Anybody else? No?
No?
Well,
Okay,
I-
well,
Catch me afterwards. then.
yeah. So join me in thanking Alicia for an amazing talk.
And but anyone who would like to join us, will go over to the well, I presume, or for lunch with Alicia. So if anyone wants to chat to Alicia about her work, very welcome to join us. Thank you.
Some of the some of the type stuff and see what happens when you go through something material.
Yeah.
Just there's only sorts of like chaotic dynamics. It was one of those things, something that happened last time.
Look at tomorrow.
Find that when we do take fire exploring, for example, depending on what type of organic matter you spike your cultures with, depends on whether you get increased cells or whether
You think you're starting to think some of these